Hi everybody, welcome to our review for HIV biology. When it comes to lectures, which one is this listed at, Sean?
What is it? HIV virus.
Okay. So we're going to start by looking through those learning objectives briefly, take a peek at them, and then we are going to kind of keep on moving. We'll come back to them at the end of a brief presentation of these slides, again, to make sure that we've covered all these points. And that'll be a good place for you to reference and make sure that you're ready from this lecture. This lecture is called HIV Virus Replication. That's what we're working off of. So let's go to the next one. We'll skip this. This was a piece that Dr. Jacobs used to talk about the difference between bacteria and a virus.
Bacteria replicate on their own. Viruses need the host to replicate. So good point to keep in mind.
Absolutely. So yeah, they're super dependent. Because they can use a host too, they can package a whole lot less. So they're much, much smaller than a bacteria. They have very limited structure, and they don't bring a whole lot of things like power plants or machinery. Really all they bring is their own blueprint or genetic material. It could be DNA or RNA. In our case, we're talking about single-stranded RNA with HIV. And we'll talk more about the structure and function of a virus, but just remember it's one of the tiniest things out there, and that they are totally dependent on host cells. This kind of just talked about that a little bit more, about relative sizes. It talks about, it plays off of the first bit Dr. Jacobs used with Phoenix being U.
Are we going to ask questions about, okay, if you're the size of Phoenix, then what is the size of a closet?
I think that wouldn't really play very well into a good understanding.
Okay, so do you know, so keep these in mind to get a general idea of what's going on, but should you be memorizing? No.
Especially if you're a non-bio major, it really helps to understand what things are. And you can look about the difference right there between the bacteria and the virus, and the virus is way smaller than the bacteria.
Yeah.
It's because it doesn't have to bring in all sorts of things like ribosomes and its own enveloped stuff.
It's a good website to check out to get a good idea of relativity with sizes between, between a the size, you, the human, to the size of your cells, to the size of bacteria, to the size of virus. Check it out on your own time. Definitely a good thing.
Good thing to look at. Cool. Got some pictures. Enjoy them. It's HIV infecting some cells. So it's very important to understand the key components of a virus. So if you're a virus, your goal, your strategy is to make more copies of yourself. In order to do that, you really just need a blueprint, which is your genetic material, because that is structurally functionally speaking, that's the most essential piece, because you need to get code in there, or your blueprint, or your DNA, in this case RNA when we're talking about HIV, into the cell, in order for that cell to make copies of it, make proteins out of there to make new virus. So when you invade, you're going to say, I'm going to hijack this cell's capability to make new copies of me as a virus, and then they're going to be able to get out of there. So that's really the overall strategy. So to do that, you need some things. You need the blueprint, like it says in point number 1 there. You need the safe, because naked, if you have your blueprint floating around, it's really easy to get damaged. So the DNA or RNA is highly reactive with other things that it would come into contact with. So in order to keep it, keep up its integrity, you have to have a safe. We'll talk about the safe a little bit. The address label, which is going to be on the outside of the virus. It's going to allow the virus to find, connect with, and then fuse to host cells. And then the few necessary tools. So when we talk about those, you might write it down now that we're going to look at reverse transcriptase, we're going to look at protease, and we're going to look at integrase as necessary tools for viral replication when it comes to HIV. A good note, these are not across the board associated with all viruses, but the necessary tools will be associated particularly with HIV.
And the necessary tools are the ones that HIV bring in by themselves. They're not something that the host cell produces. So right here we're looking at a graph of the HIV virus. We have the blueprint in the middle, which is going to be made out of RNA in HIV. Some viruses are DNA, HIV is RNA, which is important when thinking about what reverse transcriptase does. We have the safe, we have the blueprint surrounded by the safe, that's the blue proteins right there. And then surrounding those blue proteins, we have another orange protein. So these two proteins, they start out as a single protein when the virus is first made, and then protease comes and cleaves it. It cuts them in half and makes the blue and the orange proteins separate, so that we have an active virus. And we'll talk more about that here in this lecture, but pushing along. And then
when we're talking about the safe too, we're going to talk about a couple of components. So as we describe them to you, we're going to talk about the blue one as being the capsule. It's going to closely surround the blueprint, right, or the RNA, and then this one's going to be the envelope. Cool. So we have the capsule, and then the envelope, and then the outside of the envelope. Sorry, I'll just take over here. We have a lipid bilayer, so the lipid membrane. So it's important to note that the lipid membrane is picked up from the host cell, so it's not encoded by the virus. The things that the virus is really responsible for encoding for are these address labels, so GP120 or GP41. And then, like Sean said, the envelope, and then the capsule, as well as some of these special tools, like here we see reverse transcriptase. Cool. So let's talk about the blueprint. So the blueprint is small. In the blueprint, we're encoding for a small number of genes. So we talked about, again, just to recap, the genes we're encoding for are the address label, which turns out to be GP41 and GP120, the ones that we're going to talk about. We're going to talk about the capsule, and we're going to talk about the envelope, right? As well as some of the necessary tools. Because HIV, it is a blueprint, but if you looked at the color, it's going to be red. So let's just keep that in mind, because it's an RNA blueprint. So it's a red copy of the blueprint. So it gets brought in, and then in the cell, viral machinery is going to change it into blue DNA. So we start off with red RNA, and then it gets changed into like a blue blueprint. Just follow me with the analogy in the cell. We'll talk about how that happens.
So we have to go from RNA to DNA, so we do this with reverse transcriptase. Again, reverse transcriptase is a tool that the virus brings in and is not part of the host machinery. So reverse transcriptase turns out to be a very good target for HIV antiviral drugs. When you make a drug, you have to make something that's not going to attack the host, but at the same time, specifically targeting the virus, which this does very well. So reverse transcriptase, RNA to DNA, viral RNA to viral DNA, which then gets integrated into the host genome, the host blueprint, and then the host can make replicates of the virus. Yeah, that's basically all there is to it.
To Sean's point, our cellular machinery only copies DNA into proteins and into mRNA through mRNA, via mRNA into proteins. So the virus coming in says, I want to make copies of my blueprint. How am I going to do that? Well, I'm red, right? I've got this red blueprint here. It needs to be green or blue, for the host to read. And that's, just to summarize again, what he said is reverse transcriptase is going to cause that copy into DNA. The DNA then gets integrated and fixed to form double-stranded DNA.